This is an OCR A-Level Biology video on excretion. We're going to look at the whole of topic 5.2. We cover all sorts of topics. I might have to split this video down into two sections as there's a lot of content to cover here. In our previous video we looked at 5.1 and that's just a little sneak preview. At the end of it, it might be ideal to watch that video so it does build some good foundations for this topic. But let's just get started with excretion. So excretion is the removal of metabolic waste from the body, which includes CO2 from respiration, nitrogen containing compounds, so nitrogenous waste such as urea, and other compounds such as bile pigments found in feces. So anything that is a waste product of a metabolic reaction. The organs that deal with these different products, so CO2 is dealt with by the lungs, nitrogenous waste is dealt with by the kidneys, liver and skin. Skin being for like sweating, and bile is dealt with by the liver. So let's have a look at CO2 excretion. So there's two reactions here. The first one is carbon dioxide and water forms carbonic acid, H2CO3. And that reaction is catalysed by carbonic anhydrase enzyme, which I will draw on at the end. I'll draw it here, just know it's for this reaction, not this one. This H2CO3 dissociates into hydrogen ions and HCO3 minus ions. So hydrogen and hydrogen carbonate. The hydrogen ions cause a conformational change in the shape of hemoglobin. It causes it to have less affinity for oxygen, and then carbon dioxide binds to the hemoglobin. Instead of oxygen we form carbohemoglobin when that happens, and carbon dioxide then dissociates at the lungs, and that's how we can transport carbon dioxide and excrete it from the body. And the HCO3 minus at one point causes a chloride shift, but if you don't understand that process, you should, that was a bit of a whistle stop tour, you should probably watch that video on unit 3.2 which involves transporting carbon dioxide. So how do we deal with nitrogenous compounds? So in the liver we have two stages. We've got deamination, which is when we remove the amino group. So amino acid plus oxygen forms a keto acid, which is basically amino acid without the amino group, and ammonia. So ammonia is NH3. So there's that deamination, removing that amino acid, and that is the equation for it. And after deamination we get urea formation. So this ammonia and CO2 is combined to produce urea and water, and the remaining keto acid can be converted to carbohydrate or fat or used directly for respiration. So if you look at my anaerobic respiration respiratory substances video, which is unit 5.7, it shows you how keto acids can end up being pyruvate, acetyl groups, etc. They can join the Krebs cycle at pretty much any point, depending on which keto acid it is. So they have a purpose. Then this ammonia and carbon dioxide forms urea and water, and that's how we form urea. And as you'll find out later, urea is then taken out of the blood in the kidneys and then excreted from the body. Okay, so they're the two ones the textbook wants you to know about in a fair amount of detail. Bile, less detail, but we will look at it when we look at the liver. And this is our liver. And usually I speak and let the writing go as we talk, but I want to show you what we're looking at in general here. So this diagram is what I want you to see. And what I've done here is drawn a line across it, and that's sort of our cross section. And if we took that cross section and then looked down on it, we'd see this. We'd see these lobules, definitely lobules, I'm not going to say lobules, here. They're sort of hexagonal shaped, and in the centre they've got this central vein, and they've got lots of different sections to them called achini. Okay, and this is one achinus. Okay, so one of these sections, sort of like a pizza, one slice we'd call an achinus here, and at the centre of our pizza we've got this central vein. Okay, so let's get away from pizzas now. So lobules have got several achini in them with a central vein in the middle. And this is what we're looking at here. So if we take one of these slices, one of these achini, we've got the central vein in the middle here, and we've got all these branches coming off it. So first of all, we've got this hepatic artery, and the hepatic artery is bringing oxygenated blood from the heart. So it's come from the aorta, branched into the hepatic artery, and we're getting oxygenated blood from there. So oxygen flows into our achinus. Then we've got our portal vein, hepatic portal vein, and that is the vein from the digestive system, which has got all these nutrients, all these compounds, as well as a lot of waste material from digestion, and they bring it here as well, and that comes in the hepatic portal vein. So that's nutrient-rich blood. So we've got oxygenated blood and nutrient-rich blood, and then they flow into here and flow in together, and this mixing of blood happens within the sinusoid, which is sort of like a lake where lots of rivers meet. So this river from the hepatic artery and this river from the portal vein meet together in the sinusoid, and the blood mixes in there. Around here, around our sinusoid, we've got these cells, and these are our hepatocyte cells. Okay, so hepatocytes appear to be relatively unspecialized. They have a simple cuboidal shape with many microvilli on their surface. However, their many metabolic functions include protein synthesis, transformation and storage of carbohydrates, synthesis of cholesterol and bile salts, detoxification, and many other processes. I've not written all those down. One thing I did write down is detoxification, but you've got to remember protein synthesis, carbohydrate synthesis, cholesterol synthesis, and production of bile salts. Right, before I speak to you about this bile duct, we're going to look at this little cross section here. So there's a little box here cut out, and it's extended here. So in here we've got Kupffer cells, which are just move around in our blood, move about in our sinusoids, and they break down and recycle old red blood cells. And the breakdown of red blood cells produces bilirubin, which is a bile pigment, and this bilirubin ends up being transferred into the bile, and the bile is an alkaline substance which the hepatocytes produce, and that ends up neutralizing stomach acid. So bile is a secretion from the liver which has functions in digestion and excretion. The bile duct carries bile from the liver and to the gallbladder, where it is stored until required to aid digestion of fats in the small intestine. Bile all contains some excretory products such as bile pigments like bilirubin, which leave the body in the feces. So the bilirubin from broken down red blood cells enters bile. Our sinusoids are lined by hepatocyte cells. They've got these microvilli sort of structures on them, and this allows a large surface area to be produced. Blood passes along, and they remove and return substances in and out of the blood. So substances are exchanged between the blood and hepatocytes, okay, because of these microvilli, which give them a large surface area. So let's look at the detoxification properties of the liver. So there's three. I've got two here and one on the next page. So they detoxify hydrogen peroxide using catalase enzymes, and they then produce oxygen and water. They also break down drugs such as cocaine using cytochrome P450. That's another detoxification element. Let's go on our next page, which I wanted to actually play through. I'll speak to you about this liver after I've gone through this last detoxification, and that is of ethanol. Detoxification of ethanol is basically sort of a reverse of anaerobic respiration using the ethanol ethanol pathway. So ethanol to ethanol using ethanol dehydrogenase. So it's dehydrogenated, hydrogen is removed, and then there's further dehydrogenation of the ethanol to produce ethanoate, also known as acetate, which forms the acetyl group and can enter the Krebs cycle. So ethanol depresses nerve activity, but it's also got a lot of energy which can be used in respiration, and that is our ethanoate or acetate. This is a liver. This is our central vein. Sinusoids are represented here. We've got sheets of hepatocytes here, and this is really looking in a lot of detail into one lobule, and we can't see our sort of divisions of Akini. It's not that clear, but this is one lobule with a central vein, and you can't see your hepatic artery, nor you can see the hepatic portal vein. Right, so now let's look at the ornithine cycle. So this is dealing with formation of urea. And we've looked at urea formation before on the other page, where we said urea is formed with ammonia plus carbon dioxide forms urea and water. Okay, not quite that simple. There's more depth into that ornithine cycle, which is how ammonia is produced. So we've got these 3 amino acids: ornithine, hence the name, citrulline, arginine. Okay, and they all are in one cycle. So the cycle is ornithine to citrulline to arginine back to ornithine. Okay, but for these changes to occur, certain things must happen. Okay, so ammonia, which is NH3, plus ornithine, plus this CO2, creates a condensation reaction which gives out water, allowing citrulline to be produced. So ammonia and CO2 make citrulline, but water is given out as a byproduct. Then we've got citrulline, and once again ammonia, which is made by deaminating amino acid, then binds to citrulline to create arginine, but as a byproduct water is produced, so it's another condensation reaction. One water molecule, so we could just say this water molecule is then used in addition to arginine, so that is a hydrolysis. I'm not actually going to say hydrolysis because I don't know if we're breaking anything down, so just say water is added to produce urea, and then arginine turns back into ornithine. And words like this, condensation and hydrolysis, which are mentioned, but please don't write that because I don't even know if it is. The reason I'm saying this is, look, I took a picture of the specification, and this is for our exam. It says the roles of the liver in storage of glycogen, detoxification and the formation of the urea. The ornithine cycle covered in outline only. So it just wants you to know your 3 amino acids, what is added in, where, and what's produced. And the overall thing is urea is produced because ammonia and carbon dioxide make citrulline with ornithine. Citrulline and ammonia make arginine. Water is produced in byproducts in both reactions, but then water is used to form urea from arginine, which goes back to ornithine. You don't have to know how many carbons each of these things have or their chemical base, nothing like that. You just have to know the cycle. This is sort of like a general equation of what's happening here. 2 ammonia and 1 CO2 produces our urea, CONH2, plus water, 1 water, a net of 1 water. But also that equation, you can't really use equations when there's a cycle, okay, because there's more than one reaction happening. I think I'm going to pause the video there. That's been quite long. We've looked at the whole of the liver. Next video I'm going to be looking at the kidney, all of this business, which shouldn't take too long. The liver, although there was only 2 slides in it, was quite a lot to cover and quite a lot to talk about. So next video you've got a bit of kidney to look forward to.